specimen handling, transport, and processing of those samples. So proper handling of specimens is super critical. We need to make sure that we do the appropriate process for these samples to ensure that we have the right test results. So analytes may change in composition and concentration over time with certain temperature changes or exposure to light. So even if you have the best drawing technique in the world, it is going to be meaningless if the sample is not transported or processed according to the guidelines of your laboratory. So general guidelines for those specimen transport, again, this could change based on where you're working, but these are just kind of generalized ones. So the first thing is that tubes with additives should be inverted gently and completely 5 to 10 times immediately after being drawn. What this does is it helps to mix the additives so they are evenly distributed throughout the sample. If you are using a gentle inversion technique, this will also help to minimize hemolysis and make sure that we never ever mix blood together from different containers. So what that means is if you draw a green top tube and a purple tube and you have more in the green tube than the purple, you're like, oh, I'll just transfer it over. We can't do that. That will mess up results 100% of the time, and the patient will need to be redrawn. So make sure you always remember that. Another general guideline for specimen transport, so an effective and safe way to transport samples is going to be in a leak-proof bag with a zip closure. A lot of times these are going to be biohazard bags in the laboratory, and it's kind of like a sandwich bag with a Ziploc top. Specimen bags are, like I mentioned, marked with a biohazard symbol and actually have a separate front pouch for the paper requisitions to help prevent contamination of the requisition just in case that specimen leaks. Most of the time now, you're not going to have a paper requisition, especially if you were in the hospital. We have a lot of barcode labels that we are using. So, you'll label your sample with those. And if you have any extra for some reason, then you would put that in that front little pouch of the bag. Tubes should remain upright during transport as much as possible, and this accomplishes several purposes. So it helps to promote the complete clot formation when there is no additive present. It'll also help to prevent sample contamination due to prolonged contact with the stopper. And it'll help reduce the likelihood of aerosol formation during uncapping if there is no residual blood clinging to the stopper. There are time constraints as mentioned as well. So the quality of test results is going to depend a lot on the time between when the sample is drawn and when it is analyzed. The longer the interval, the more likely the results will actually be inaccurate. The ongoing glycolysis, which is the metabolic sugar breakdown within cells within the specimen, is a primary cause of inaccurate test results because it continues even when it's in the tube. So it's going to continue to break that stuff down, and that is actually what we're trying to test for. So that will then give us bad results. So as a general rule, an uncentrifuged blood sample should be delivered to the lab within 45 minutes of being drawn. There are many different types of tests that can be affected by glycolysis, which include glucose, potassium, calcitonin, phosphorus, aldosterone, and a whole bunch of different enzymes. So according to CLSI, it says 2 hours should not pass between collection and separation of centrifugation of cells from plasma or serum. But like I mentioned earlier, we kind of usually say, 45 minutes or less for sure. To get it to the laboratory. A few sample types can wait longer before processing without loss of viability. That's because fluoride will inhibit glycolysis. So those glucose samples that are collected in gray top tubes can actually be held for 24 hours at room temp and 48 hours at 2 to 8 degrees Celsius. So if for some reason it was a glucose and we had to wait, we couldn't refrigerate it or we couldn't, get it to the lab in time, this is an alternative, although it won't happen very often. But if you needed it to, you could. Blood smears that are made from these samples must be done within an hour of collection because the EDTA additive will eventually distort the cell morphology. And that is the tube type that we are going to use to try to determine cell morphology. So we need to make sure that we get it there in time for that as well. So some temperature considerations for specimens. So keeping those specimens warm. Specimens that must be maintained at 37 degrees Celsius or body temp during transport and handling will include the cold agglutinins that we mentioned, cryoglobulins, and cryofibrinogen. These tubes for these specimens should be warmed using a heel warmer packet before collection, and the sample should be transported wrapped in a heel warmer packet as well. So heel warmers are actually effective for up to about 30 minutes. And then the phlebotomist should alert the lab staff of the arrival of the warm sample to make sure that the people in the lab are aware that it's there, and then they can make sure that it's held at the right temperature until it is tested as well. Keeping the specimens at room temp. So some specimens must be kept at room temperature. Make sure you follow your facility's protocols for placing specimens in a transport device for delivery because they could be different. But if the specimen is being delivered outside the facility by a courier, it may be placed in an insulated container, which will actually help to protect it from extreme heat or cold. All right, keeping those specimens cool. So sometimes a specimen needs to stay cold, and that actually helps to slow the metabolic processes and keeps analytes stable during transport and handling. Samples that need to be chilled will include pyruvate and lactic acid. To chill a sample, we're going to place it in a slurry, which is basically ice and water, and that helps to promote complete contact between the sample and the ice bath. You will want to avoid large ice cubes, so that's kind of why it mentions the chipped ice, just because it could cause the sample to freeze if you're using the large ones. Keeping those specimens frozen. So some specimens need to be kept frozen because some analytes are only stable in extreme cold. They will require storage at temperatures between negative 20 degrees Celsius all the way down to negative 70 degrees Celsius before testing can occur. And this would be for certain things like clotting factors and complement factors. So we're always going to freeze in plastic tubes that are actually designed for low temperatures and use labels that can adhere to plastic at low temperatures as well. Tests that require freezing often require special equipment or reagents and are usually sent out to reference laboratories and must be transported frozen, and we do that by using dry ice. Protecting those specimens from light. So exposure to light can break down light-sensitive analytes. Like we mentioned before, bilirubin is the most common light-sensitive analyte, but there are some others such as vitamin B12, carotene, folate, and urine porphyrin. To prevent the light exposure, samples are going to be collected in those amber-colored tubes. They can be wrapped with aluminum foil or an amber-brown biohazard bag as well. And then they're going to be placed inside, an envelope or heavy bag or a cooler or something, which also helps to keep it protected from light while it is being processed and shipped. Transporting specimens to the laboratory. So in a lot of institutions, samples are going to be hand carried by the phlebotomist or another member of the laboratory team to the laboratory. Samples may be dropped off at designated areas within the hospital for transportation and delivery by lab staff, and then minimum information should include the patient's name, hospital number, and room number, specimen type, date and time of delivery to the drop-off area, and name of the person depositing it. Some institutions, such as hospitals, generally will have a pneumatic tube system, so kind of like the bank when you go there and you put your money in and it goes up and the tube thing comes back, and that's basically where the samples are carried in that sealed plastic carrier that travels within the network of tubes. It has shock absorbing foam inserts that are placed in the carriers, and that helps to reduce the shaking and any agitation of the sample during the transport. There are special containers that are used to protect the sample and prevent contamination of other material during transport. Pneumatic tube systems are often used for the delivery of paperwork and other items, but are not always appropriate for blood samples, and the lab needs to assess how well the system meets its needs. So some larger laboratories may have self-contained motorized carriers that run on a track between departments within the lab, and that actually helps to transport the specimens within the lab as well. Safety, so the Occupational Safety and Health Administration, known as OSHA, requires personal protective equipment or PPE to be worn during sample processing. The required equipment is going to consist of gloves, a lab coat, buttoned or snapped closed with closed cuffs, Protected face gear, so either goggles or a mask or a chin-length face shield. So it kind of depends on what you're working with and what you're doing and what your facility's protocols are for what you need to be wearing while you are processing samples. So central processing, specimens enter the lab are usually first handled by an area called central processing. And this is an area that's devoted to accessioning and sorting samples as they arrive. So the date and time of the sample is recorded with a time and date stamping machine or recorded into the computer via barcode. Each sample is marked with an accession number, a unique identifying number that is used for cataloging the sample in the lab. Like I mentioned earlier, samples are labeled with barcodes that are read by an electronic reader, which also records the time the sample is received and then stores that in the computer system. Samples are then sorted by sample type and then transported or divvied up within the laboratory. Central processing is also usually responsible for centrifuging those samples, and that is where they separate plasma or serum from the cellular elements of the red blood cells. They also help for preparing aliquots, which are small portions of the specimen that are then transferred into separate containers for distribution to different laboratory departments. And then before centrifuging, the stopper should remain on the sample to prevent its contamination or alteration. So with that, if you remove the cap, it actually releases carbon dioxide, which raises the pH in the sample and allows sample evaporation, which can then cause an increased concentration of analytes, so it can give you bad results. An open tube is also likely to pick up dust, sweat, powder from gloves, or other contaminants. It also creates the possibility of infectious aerosols during centrifugation as well. So clotting, serum specimens must be completely clotted before centrifugation can occur. Complete clotting can take 30 to 45 minutes at room temperature. Samples with clot activators, including serum separator tubes, will usually clot within 30 minutes. If thrombin is used, the complete clotting may occur within 5 minutes. Incompletely clotted samples continue to clot after serum separation, which interferes with testing. And samples from patients on anticoagulants like heparin or Coumadin have longer clotting times, as do chilled specimens and those from patients with high white blood cell counts. So centrifuging. A centrifuge spins the sample at a very, very high speed, and that actually separates the cellular components based on density. So you have usually your red cells on the bottom, and then in between, on top of that, you will have your platelets and your white cells, and then on top of that is your serum or your plasma. So the cellular elements, which are denser, like I mentioned, the red cells move to the bottom, and then that less dense plasma or serum is actually pushed to the top. Centrifuges come in a variety of sizes, from small tabletop models designed to hold only 6 to 8 specimens to big, large floor models that can hold, 20 or 200 or more. The failure to balance the load causes the rotor of the centrifuge to spin out of center, and this can damage the centrifuge and may allow it to move during operation, which could possibly cause it to fall off the floor or move across the floor. Tubes must be balanced in the centrifuge to avoid creating a hazard and damaging the machine. So removing a stopper from the specimen, the major risk of stopper removal is the formation of an aerosol, which is a microscopic mist of blood that forms from droplets inside the tube. Aerosols are especially likely if the tube or rim has been contaminated by blood during collection or transport. And many automated instruments allow for testing without stopper removal, so that means the lid can actually stay on a lot of these newer instruments coming out now. So when you are removing a top from a tube, always use a safety shield to prevent blood from accidentally splattering on you and to reduce the risk of aerosols. A personal shield has a headband that sits on your head kind of like a hat or like a visor. So it has like that clear plastic visor that you pull down over your face. And then a workstation shield attaches to the work counter from either above or below, and that also is there to help protect your face from any splattering. And you can usually adjust its height to offer the best protection to whomever is using it. Preparing those aliquots. So all tubes into which aliquots are placed should be labeled before filling and then capped before delivery to the appropriate department. Aliquots are not poured off because this may cause splashing and aerosol formation. So instead, we actually use a pipette to pull off some specimen and put it into another container. Aliquots may also be prepared automatically by an instrument in the lab. The system may include automated centrifuging and delivery of samples to testing instruments. Again, it's just going to depend on where you're working. To prevent an identification error, you must label the transport tube before aliquoting the sample and then carefully match the patient information on the original tube with that transport tube. So making sure that you're never taking a sample out of a tube and putting it into a tube that is not labeled. Because you might get pulled away. You might go answer a phone and then someone would come in and they could potentially label it incorrectly. So make sure you always label your tubes first when you're preparing aliquots. Transport and processing of non-blood specimens. So microbiology samples must be transported to the lab immediately, and that helps to increase the likelihood of recovering any pathogenic organisms. Most specimens are collected in transported media and do not require additional processing after collection. And most samples are plated immediately on culture media. Often the phlebotomists are going to be trained on how to perform this task. So depending on where you work, you may or may not actually do some plating for microbiology. Specimen rejection. So all specimens received by the lab must be evaluated for acceptability before further processing. So these are some reasons why specimens would be rejected. So like we mentioned before, if the sample is hemolyzed, if we have the incorrect tube that was ordered for the test, so let's say we need a CBC and we needed a lavender tube, but we received a green tube. We can't do a CBC on that, so it's going to be rejected. Drawing a specimen at the wrong time could cause a specimen to be rejected, and also improper handling. So if it took too long to get to the lab, things like that could all cause rejections of specimens.